beginning of the COVID-19 pandemic, the World Health Organization had a simple message to the world: test, test, test. This means that knowing whether a person is infected or not is critically important in fighting off and containing the spread of COVID-19.
Even with the country's initial success in sourcing and producing diagnostic kits, expanding the reach of COVID-19 testing remains a challenge. Aside from increasing test accessibility, we also want to encourage more people to undergo testing when needed.
Some of the challenges for mass testing include tedious procedures that lead to longer waiting time for patients, uncomfortable nasal swabs, which can be very daunting to some, difficulty in test deployment because of the required technical expertise and huge expensive machines, and lastly, costly tests. To further strengthen the country's capacities, we turn to science for technologies that can make testing faster, cheaper, and simpler.
This is why we, the Bio and Biomedical Research Group from the Ateneo Research Institute for Science and Engineering, or ARISE, launched Amplified. Amplified combines 3 independent projects with a shared pursuits and end goal of amplifying COVID-19 testing capacity in the Philippines. The first project is the miPCR, a Microfluidic PCR Device. The 2 other projects that enable saliva-based detection of the virus are ADS, a lateral flow DNA detection system, and Swifter, development of isothermal COVID-19 testing technology. These 3 DOST PCHRD funded projects each create innovative testing products. But to bring COVID-19 testing closer to all Filipinos at the point of their need, both ADS and Swifter are planned to integrate with the miPCR system. As a team leader, I will tell you more about miPCR. So what is the miPCR? A PCR machine is the gold standard instrument used to find out whether patients are infected by a pathogen. However, it is difficult to deploy and distribute because the equipment needed is large, complex, and expensive. To address the problem, we designed the miPCR, a device roughly the size of my palm which has the functionalities of a PCR. So how are we able to compact a large PCR machine into this compact device? Using creative electronics engineering, our team designed a unique and simple heating system and combined it with a cheap microfluidic testing cartridge that we designed. When optimized, cartridges use only very small amounts of patient sample and test reagents, and simplify test preparation, further cutting down the cost of a COVID-19 test. Because of the design of the miPCR, we project the final device to be at least 5 times cheaper than current qPCR devices. The smaller, more compact, and cheaper miPCR will also make distribution much easier, allowing us to reach even community clinics with limited laboratory capacities and limited budgets. By enabling testing closer to the point of care, patient samples can be processed locally, removing the need for slow, expensive transportation of patient samples to a main testing lab. Results can be processed faster, and patients and healthcare workers notified sooner. With the miPCR composing the engineering component of the amplified testing system, how about viral detection? ADS and Swifter have the answer to that.
Typically, COVID-19 testing requires a nasal swab. The swabs are first prepared before they proceed to the actual PCR testing. This has data and results looking like this. This entire process is time-consuming. It also requires technical expertise to perform the test and interpret the results. ADS and Swifter make tests simpler by replacing swabs with only 3 steps. Step 1. Collection of patient saliva. Step 2. Heating the saliva collected. And Step 3. Detection of the virus in saliva using cheaper and less complex methods. Why saliva? Saliva can be easily and quickly processed by a medical technician or almost anybody. More importantly, since saliva can be self-collected, we now have an option to avoid the uncomfortable nasal swabs. The main difference between the two projects lies in the technology used. ADS retain the PCR technology but simplifies the readout. Instead of looking at graphs, ADS makes use of a cheap lateral flow device or a dipstick. It is very much like a pregnancy test, but for COVID-19. By simply looking at the lines, we will know whether they are COVID positive or not. Swifter, as the name suggests, aims to have a faster turnaround time of results and hence shorter waiting time. Swifter uses a simpler effective version of PCR called LAMP, which runs for only half an hour. Currently, with qPCR testing, delivering the results to patients take at least 6 hours. With Swifter, the waiting time can go down from 6 to just within 1 hour. With the LAMP technology, the development of test kits that enable rapid, simpler, and more cost-efficient testing is made possible. Through miPCR, ADS, and Swifter, we seek to enable more COVID-19 testing in more primary care clinics and hospitals by making saliva-based testing more financially accessible for more Filipino patients.
While designed to work with typical PCR instruments, ADS and Swifter tests are designed to function with our miPCR device as well. The benefits of Amplified are maximized when both the miPCR and the ADS or Swifter testing kits are used together. For ADS, instead of buying a new thermocycler, the miPCR can be used with the ADS kit to enable scaled and distributed testing. Similarly, the miPCR can be used to run the Swifter LAMP test for both the amplification and test readout. The Microfluidic cartridge can also be configured to allow for multiple samples to be tested in a single run.
Our government has always emphasized the 3 T's: trace, test, and treat in its fight to stem the spread of the virus. However, some of the major constraints that we face during this pandemic are the high cost of testing for COVID, and even worse, the lack of testing facilities or devices in various locations in the country, especially in the more remote rural areas. Many LGUs cannot afford the financial burden of building a biosafety level 2 facility in order to do this testing. And so, if we are to save more people from dying from COVID-19 due to their lack of access to faster and cost-effective tests at the barangay level, if we want to send our children to school with the assurance that school clinics are capable of conducting widespread testing for all their students, the faculty, and the staff, if we are to save the economy by providing our workers with a safe and healthy working environment, then we need to be able to make these devices as widely available as possible at reasonable costs and at the soonest possible time.
We cannot fight the virus if we do not know who is infected. We need to test, trace, and treat. The timing of the test is vital to the success of fighting off any disease, both for each person and in our communities. The earlier, the better. Our Bayan projects will not stop with COVID-19. The technology we develop can also easily be applied to other common Philippine diseases like tuberculosis, malaria, or even cancer.
We hope that the Amplified project will expand coverage of COVID-19 testing for the Filipino people, and everything we have learned can be used to accelerate development of diagnostic tests for other diseases in the coming years.
Faster, cheaper, simpler. Better testing by Bayan, para sa Bayan.